So I'm going to switch gears, and we're going to talk about a genetically heterogeneous disease for which structural variations are tremendously important. So this disease is congenital diaphragmatic hernia. It's one of the most common structural birth defects, and I'll tell you a little bit more about it. So CDH is how I'm going to be referring to this disorder throughout this talk. Is a birth defect which results in incomplete formation or muscularization of the developing diaphragm, which is, of course, our major respiratory muscle. And this results in displacement of the abdominal contents into the thoracic cavity. The main problem with these babies is that they have significant lung problems, including a comorbid lung hypoplasia and pulmonary hypertension. And this is really what kills them, unfortunately. It is one of the most common structural birth defects. It has an incidence of up to 1 in 2,500 live births, and it is quite severe with a 50 to 80% survival rate and significant morbidity and mortality despite advanced surgical and medical care. Interestingly, about 60% of these patients have isolated diaphragm defects without any other congenital anomalies or any other health concerns. So we really think that if we can fix their diaphragms and prevent their lung disease, that we can portend a really good clinical prognosis. But we understand that transformative treatment strategies are going to require an understanding of basic developmental mechanisms. So there is significant support for a genetic etiology of this birth defect, along with a number of other structural birth defects. So there is an association of several single gene or chromosome syndromic disorders with CDH. There is an observation of families with more than one affected individual. I'll show you guys an example of this. There's also presence of CDH in mouse genetic models and a growing number of CDH-causative genes and genetic mutations in human patients. As I mentioned, the genetics of this disorder are highly heterogeneous. There's a long-standing history of studying structural variations in this disorder. At least 10 to 15% of cases of CDH are associated with chromosomal abnormalities, including aneuploidies and recurrent deletion hotspots. So these have all been studied by classical cytogenetics and then chromosome microarray technologies over decades. There are a number of these recurrent rearrangements that I've noted there. But I think that these are really this is just the tip of the iceberg. I think that we're finding that there are actually many more structural variations beyond this that are responsible for this disorder. Importantly, these associations have led to the identification of important CDH genes, and systematic analyses of structural variants in large cohorts has been lacking. Most of these are just single case reports or small case series. So myself and my colleagues have been recruiting patients with this birth defect for over 17 years at Mass General Hospital, Boston Children's Hospital, and through referrals and patient support groups. We currently have about 650 individuals affected with CDH enrolled in our study and about 1,200 unaffected family members. And we've also joined forces with the DREAM study, which is a multi-site study led by Wendy Chung at Columbia. And by combining our studies, we have been able to increase our cohort size to over 1,500 patients. This is actually the largest cohort of CDH patients in the world. And we have performed whole exome sequencing and whole genome sequencing for a total of about 800 trios. So these are patients with their unaffected parents. And we are currently analyzing structural variants in a large cohort. So we've been using several different strategies to assess structural variation in this disease. We and others have published a number of cases using array comparative genome hybridization. And then, of course, the focus of today's talk is going to be using the optical genome mapping technology. And then we're also in the process of analyzing sequencing data, either whole genome sequencing or whole exome sequencing for structural variation. So I'll just tell you about the study design that we used for the study using the Bionano technology. So we chose samples with a mix of patients with either known or suspected structural variations. And we also had a number of unknowns for whom a causative genetic variant had not yet been identified. Our goals were really threefold. First, we wanted to confirm these known structural variations and see if we could gain any additional insights into higher order structure to help refine the pathogenesis of those structural variants. We wanted to see if we could identify novel structural variants, and we wanted to compare the structural variant calls with different technologies. So this was just a summary of our patients. We did 5 trios. These were patients with unaffected parents in order to find any de novo structural variants. We had 12 singletons, and then we had 2 patients from multiplex families. So these are families in which more than one individual in the family was affected with CDH. About half of the patients had a previously identified structural variant that had been detected by an ASHG technology or karyotype, and about half were basically unknowns. Before I sort of launch into some of the examples, I just want to summarize our results. We confirmed all the high-confidence structural variations that were detected by ASHG and karyotype, and we provided an additional level of detail for several structural variants. And we identified at least one novel likely causative structural variant as well. So the first example I want to share with you, this was from one of the trios. This is actually a de novo balanced translocation that had been previously identified by karyotype, but we really wanted to try to delineate the breakpoints of this balanced translocation in more detail. So this is a balanced translocation between chromosome 1 and chromosome 6. Here it's depicted. And using the Bionano technology, we were able to identify the breakpoints for both of these chromosomes. The breakpoint on chromosome 1 wasn't terribly interesting to follow up on, but the breakpoint on chromosome 6 was quite intriguing. So it's upstream of a gene that's called VGLL2. And VGLL2 is a gene that functions as a transcriptional coactivator of myogenic differentiation. This was quite intriguing to us because, of course, the diaphragm is a muscle, and we know that many CDH-associated genes are a part of the myogenic differentiation pathway. What was interesting is this breakpoint actually is between VGLL2 and an upstream, very highly conserved element, which, if you look at the encoded data, seems to have marks of so DNase I hypersensitivity and an open chromatin structure that's prominent in muscle cell lines. So this leads us to hypothesize that VGLL2 might be an interesting candidate gene to follow up in further functional studies. The next example I want to share with you is a rearrangement at 16p11.2. This was actually a rearrangement that we had identified using ASHG technology as being enriched with statistical significance in patients with CDH compared to controls. So this is actually that's found at a low level in the control population or in general population. So we hypothesized that it's a risk factor for CDH. And by ASHG, this appeared to be a comet, just a single duplication of 2.3 megabases. But when we performed Bionano on this sample, we actually showed that there's a considerably more complex rearrangement with 2 adjacent smaller duplications, one of which was inverted. And this is just the output from the Bionano Access software. I've also sort of depicted this graphically here as well, so you can see. So there are these 2 regions in purple and orange that are both duplicated, and this one that's in orange is also inverted and inserted upstream. So this really gave us a considerably more information about the complex structure of this rearrangement. It identified additional candidate genes. And what's interesting about this region, as has been noted, that this technology is helpful for studying regions with extensive segmental duplications, and that is in fact the case in this region of 16p. The next case I want to share with you is a multiplex family. So CDH does occasionally run in families. And this is a family that was shared by a collaborator of mine in France, Florence Petit. So this is a family that was found to have a copy number gain of the X chromosome, Xq13.1, that involves the FRMB1 gene. So for any clinicians in the room, you might know that FRMB1 is causative of a genetic syndrome called craniofrontodysplasia. This is an X-linked dominant condition. It's not typically associated with CDH. About 10% of patients have CDH. But what's interesting is that there are these 3 cases that have been reported with copy number gains in this region that appear to cause a distinct syndrome with a high penetrance of CDH. And so this family with these affected females, all of these females have CDH. And then there are 2 other families that have been reported, or 2 other cases that have been reported. So we wanted to understand this copy number gain in a bit more detail. So this was quite intriguing. When we actually used the Bionano technology, what appeared to be a duplication by array CGH actually turned out to look more like a triplication. So you can see this here on the top. This is the copy number profile read from the software, and you can see it's 3.8 copies, consistent with a triplication. And then when we do the alignments, what you can see is not only is there a tandem triplication, but that one of these reads is actually inverted. The next case I wanted to tell you about was a novel structural variant that we found in this cohort. So this is one of the singletons that we studied, and we were able to identify a complex rearrangement on chromosome 15, which resulted in a deletion, which is depicted here, including the NR2F2 gene, and then an adjacent inversion. This is just depicted here again. So this includes a deletion of the NR2F2 and NR2F2 antisense transcripts, and then in this inversion adjacent. I'll talk to you a little bit more about what NR2F2 is, but there was another case that was in our cohort, which was an unbalanced translocation that we also studied using the Bionano technology. And this unbalanced translocation resulted in a large distal deletion of 15q, which also included this gene. So NR2F2 is a transcription factor. It's otherwise known as CoopTF2. It's a co-repressor of the retinoic acid signaling pathway, and retinoic acid signaling is one of the key core components of diaphragm and lung differentiation. And there are multiple genes in this signaling pathway that have been implicated in this disease. This is a known CDH gene, and we and others have shown that de novo mutations are identified in patients with CDH. It's also linked with congenital heart defects, and mouse models show a role for this gene in CDH. So this technology did identify what we believe to be a pathogenic structural variant in one patient in our cohort. So to conclude, I just want to make the point that structural variants are important for the pathogenesis of congenital diaphragmatic hernia. I think that this model can actually be expanded to study other genetically heterogeneous structural birth defects, which would have a similar type of pattern. And that the optical mapping technology using the Bionano platform provided an additional level of resolution of some of the structural variants that we saw compared to the array CGH. Then we think that analysis of structural variation and sequence variation in parallel is going to lead to identification of critical genes and pathways for this disorder. And direct comparisons of the different platforms for assessing structural variation will be useful to determine which technology or combination of technologies will be ideal. So we have a number of future directions for this project. Upcoming, we are hoping to use this technology on a larger discovery cohort we have ready to go of 50 patient-parent trios, and we're hoping to use this to identify novel structural variants. We are also in the process of analyzing structural variants from whole genome and whole exome sequencing data. And many of the patients enrolled in our study have now had testing both by Bionano as well as genome sequencing and/or array-CGH, and we plan to directly compare the calls using these different technologies. Just want to stop by thanking my team in MGH, also my team at Boston Children's Hospital, and Florence Petit from the Hôpital Jean de Flandre, sounds fabulous, who provided the FMB1 family. I want to thank the Bionano genomics team, especially the bioinformaticists who have been helping me learn how to use the software and do the analysis for these samples, and my funding sources.